In Dr. Leavey's Module 3 of Bio 1510 at Georgia Tech, we've been learning about cellular biology, from molecules to membranes to metabolism. Our group, B5, was assigned to discuss and present a case study on the differences in cellular metabolic rate in two distinct types of birds. In this study, researchers from Ohio State University and the University of Cincinnati hypothesized that there was a correlation between the metabolic rates in the cells of these animals and their overall metabolic rates and proceeded to test them. Researchers collected tropical and temperate birds in two locations. The tropical birds were collected in a lowland forest in Gambia, Panama, and the temperate birds were collected around Ohio in the U.S. To obtain the cells from the birds to culture in the lab, the researchers first dispatched the birds and then collected skin samples, which were then cultured in the laboratory. The birds from Panama were sent overnight in order to minimize the transition time. Also, even though the birds were collected from geographically different locations, the scientists took every effort to ensure that the climate of both regions was fairly similar, with an average air temperature difference of only 4 degrees Celsius. The birds from Ohio were collected in May and June in order to achieve this minimal temperature difference. There were 17 species of birds collected from each of the two locations. Each species' cell samples were tested for the oxygenic consumption and glycolytic capacity. Immediately after the bird's death, a 25 square millimeter piece of skin was excised and cleansed. After being left in a solution overnight, they were grown in an atmosphere of 5% oxygen at 37 degrees Celsius. After day 3, the solution was replaced, and from day 7 to 10, the cultures were placed in new flasks. A week passed, and the cells were harvested and stored in liquid nitrogen for up to 12 months before their metabolic profiles were assessed. Before the assessment, all the cells were thawed and allowed 5 days to recover from the treatment before they were tested. A device called a Seahorse XF24 extracellular flux analyzer was used to measure the rate of oxygen uptake and carbon dioxide output in these cell cultures. Each culture was placed in a running medium containing glucose and sodium pyruvate and was allowed to acclimatize for an hour before it was tested for oxygen consumption and metabolic rate. This experiment takes many of the course concepts we've covered and puts them into practical use. In this experiment, researchers performed paired comparisons of cellular respiration between two different subgroups of birds from two geographically different regions. We defined cellular respiration to be the set of metabolic rates and processes that converts nutrients to ATP in an organism, as Dr. Leavey has taught us. The study of cellular respiration is a fundamental part of this case study. These groups included all shapes and sizes of birds, with masses ranging from 3 grams to 327 grams, with an aim of determining whether species that live so-called fast and slow-paced lives have correspondingly fast and slow-paced metabolisms. It was noted that, across the species, the actual sizes of the cells were not significantly different. Also, basal cellular oxygen consumption, or the minimal amount of energy required to maintain vital functions in an organism at rest, was an average of 29% lower in the tropical bird species. While not explicitly mentioned in class, basal cell oxygen consumption is an important part of cellular respiration and understanding how cells function as a whole. Another phrase of note in the context of the study is the concept of proton leak, or the movement of protons across the inner membrane of the mitochondria, as we have also learned. Just like with basal cellular oxygen consumption, proton leak was significantly lower in tropical birds. These findings were in keeping with Breann's 1999 research, which showed that species with fast life history exhibit rapid development, intense metabolism, high reproductive rates, and reduced longevity, while species with slow life history show the opposite pattern.
What you see on the screen before you are 4 different graphs displaying the differences between tropical and temperate bird values for each of these 4 criteria, which I've just talked about. The dotted line on there represents the sort of limiting factor between the two groups, and since most of the values are in the negative range, as explained in the figure description, this means that most of the values for the tropical birds were lower than the values for the temperate birds.
As shown by this experiment, the basal metabolic rate is inversely proportional to the pace of life of the organism. That is, the more offspring that an organism produces in its life, and the shorter life it lives, then the higher its metabolic rate will be.